Hi class, Miss Aarons here. Today we're going to be talking about 8.3, which is going to be chromosomes and DNA replication. After this lecture, you should be able to summarize the events of DNA replication, and you should be able to relate DNA molecule to chromosome structure. A perfect copy. Here is a little pre-activity that I want you to do. When a cell divides, each daughter cell receives a complete set of chromosomes. We know that from mitosis and meiosis. This means that the new cell has a complete set of its own DNA code. Before a cell can divide, the DNA must be copied so that there are two sets to be distributed into new cells. So what I want you to do is, on a sheet of paper, I want you to draw a curving or zigzag lines that divide the paper into two halves. You can make a vary between the bends and the lines as you draw it. Then, without tracing, copy that line on a second sheet of paper. Now I want you to hold the papers side by side and compare the lines. Do they look the same? Now stack the papers on top of one another and hold the papers in the light. Are the lines the same? How could you use the original paper to draw the exact copies without tracing it? Why is it important that the copies of DNA that are given to the new daughter cells are exact copies of the original? So here's a little bit of a review for you. Prokaryotic cells have no nucleus, so the DNA is located in the cytoplasm. And we have an example right here for you. This is the DNA. It's located in the cytoplasm of the cell. A single DNA molecule contains all of the genetic info. And here we go, here's the DNA. The strand of DNA. Eukaryotic cells are going to be more complicated. The DNA is located in the nucleus and has chromosomes. DNA length, it is going to be incredibly long, and the nucleus of a human cell contains more than a meter of DNA. So you know, like the meter sticks, the big long sticks, it contains more than that in the little cell. So it's very, very tightly coiled and very small. Eukaryotic chromosomes contain both DNA and histones, which are the proteins. The chromatin consists of DNA tightly coiled around a protein called the histones. DNA and histone molecules are like bead-like structures called a nucleosome, which are these. Nucleosomes are packed with one another to form a thicker fiber. So then we have the coils, and then we have the supercoils. Shortened by the system of loops and coils. So that's what makes it shorter. So we have a little bit of naming here. We have the DNA, then we go to the histones, then we go to the nucleosomes, then we go to the coils, then we go to the supercoils, which gives us the chromosome. So DNA replication. Each strand of DNA is a double helix and has all the info needed to reconstruct the other half by base pairing. Guanine goes with cytosine, adenine goes with thymine. And it's always this way. C goes with G, G goes with C, A goes with T, T goes with A for DNA replication. Replication is going to be the process that creates a copy of the DNA. So the DNA molecule unwinds at the replication fork and helps the enzyme helicase So helicase actually unwinds a DNA molecule. And that's when the hydrogen bond is broken between the base pairs. The two complementary strands using base pairing, and the new DNA molecule has one original strand and one new strand. So they unzip the DNA, and then using helicase, the complementary strands start binding with new strands to make two new sets of DNA. So here's an example. The enzyme for DNA replication is called DNA polymerase. And it joins individual nucleotides to produce a DNA molecule. And it proofreads the DNA, so it's going to make sure that the right things are bonded to each other. So here we have our nitrogen bases. We have our replication fork. We have the original strand right here, which is the blue. And then we have the new strand here, which is the orange. The DNA polymerase is right here, and that's what's forming the new bonds for the new strand and the old strand. And that's kind of how it works. To go more in depth here, I want you to turn to your book and look at page 223. It actually talks about the replication process as well. So I'm going to go ahead and switch to a new PowerPoint and go into that further for you as well. Okay, here's PowerPoint number 2. So to go into it a little bit further, like I said before, proteins carry out the process of replication. DNA is only going to serve as a template. Enzymes and other proteins will actually do the work of the replication. The enzyme will actually unzip it, and the free-floating nucleotides form hydrogen bonds with the template strand. So here we go. So right here, we have it unzipping, and then we have the new nucleotides here forming together. DNA polymerase, which is the enzyme that bonds the nucleotides together to form a double helix. The polymerase enzyme will form covalent bonds between nucleotides in the new strand as well. So we have the old strand right here, which is the blue, and then the orange is going to be the new strand. And C is going to bond with G, and A is going to bond with T. So you're always going to see yellow with purple or green with red. And that's how it's always going to be. The new molecules of DNA that are formed, each with original strand, are going to form a new strand. DNA replication is semi-conservative. So what it's going to have is, now you saw that the two things forming, now we have two whole new strands of DNA. So one's going to go to one cell and the other one is going to go to the other cell. Replication is normally very, very fast and very accurate because the DNA polymerase will actually check it as well. The DNA replication starts at many points in the eukaryotic chromosome. And the DNA polymerase will find and correct errors, like I already said. That is the end of my video. If you have any questions about DNA replication, don't be afraid to ask.